So this video is about the DNA replication errors. Human beings obviously have 23 chromosomes. The chromosomes carry DNA, which hold the instructions for our growths for the body and our functions. Every time a cell divides, it must copy all of the DNA. Even though the DNA replication is very precise and accurate, there are obviously there can be errors, and these errors can have important consequences for human physiology and just different types of disorders that we know of today. So the errors that I found for DNA replication, so the DNA copying enzyme, the DNA polymerase, it sometimes makes mistakes, and there are different types of mistakes, and the mistakes are called mutations. There are different types of mutations. I'm reading off my notes that I actually took in A&P, so this is actually a great topic. The examples of different mutations are point mutations, insertions or deletions, and large errors. The point mutations, it's basically where one base is swapped for another base that kind of doesn't belong there. Insertions or deletions is basically the addition of extra bases or like lost bases. And then there are large errors where the whole sections of the DNA is either duplicated or it's just missing. And then the consequences of these errors that I just mentioned. So if the errors remain, they can affect how our proteins are made and how the body will function and grow. Some examples that I found trying to relate to our society today are Down syndrome. Down syndrome is linked to errors in a chromosome separation, leading to an extra copy of chromosome 21. Also a big one in today's society, and it's always been a big one, is the cancer. It's caused by mutations in genes that control our cell growth. And then another one that I listed, I'm not too familiar about, but I found this in my article, is cystic fibrosis. It's often caused by a deletion of 3 DNA bases, going back to the insertion or deletion errors in my replication category, which leads to faulty chloride channels in the cells. And then this was a big one that actually football players that I played with, when I actually even played here my first year, everybody got tested for it. It was the sickle cell anemia. It's caused by a single base change in the hemoglobin gene, basically changing the red blood cell type. So it's out of its original shape. I'm sure everyone knows the shape of a red blood cell. And then for what I got for my articles from DNA repair mechanisms, they have different types of repair systems that can fix most of these errors, though not all the errors can always be fixed, I don't think, that from my research. So some repair systems that I found were proofreading by DNA polymerase. Just basically, when there is a mistake, the proofreading by DNA polymerase basically analyzes, checks, and removes mistakes during copying before the copying process is complete. Another one is a mismatch pair. It basically replaces incorrectly paired bases, going back to where I talked about my insertions or deletions of extra bases added or lost. This basically replaces the addition or missing pair of the DNA repair bases. And then the last one was nucleotide excision repair. It removes larger damaged sections, such as from UV radiation. I didn't know too much about that, but I found it interesting in my article. I actually touched base with my boss at work. I work at PT Solutions, and he said that that was a big one they actually studied in grad school. So that's why I listed that one. And then the big question is, do enzymes fix all errors? I don't. My personal opinion, I don't think they do. But what I found was enzymes in the cell fixed most errors, but like I said, I don't think they fix all of them because some mutations slip through and become permanent. Hence why some of the disorders or consequences that I listed are still standing today, and they become permanent. While some of them are harmless, many others of them do cause diseases, like the cancer that I mentioned or genetic disorders. Rarely a mutation may be helpful in a way, contributing to genetic diversity and evolution. So without the enzymes fixing all these errors, we wouldn't really know, we probably wouldn't know about the different types of consequences and the evolution of these errors. So in conclusion, I do think DNA replication is very accurate. However, there are some situations where the enzymes cannot correct some of the mistakes, but they do correct most of the mistakes. And then going back to where I listed the different types of consequences, these permanent mutations are what lead to the sickle cell anemia, the cystic fibrosis, the cancer, the chromosomal conditions. Although they're harmful in many cases, replication errors also play a role in human adaptation and evolution. So, yeah.